I would now like to do a quick deep dive from the epidemiological data which was mentioned in the YouTube Sunlight for Hospitalized COVID Patients, namely the latitudinal dependence in COVID mortality, and bring that down to the subcellular level, particularly mitochondria and peroxisomes. So in this YouTube, Professor Schwalt was discussing this key paper, Ultraviolet A Radiation and COVID-19 Deaths in the USA with replication studies in England and Italy. And this paper was published in 2021. This epidemiological data needs to be connected to ferroptosis biomarkers within cells, particularly cells that have been treated with sunlight or near-infrared light. And that brings us to this paper on the right. Near-infrared light exposure triggers reactive oxygen species to downregulate inflammatory cytokines induced by SARS-CoV-2 spike protein in human cell culture. Now, this paper is complex, but it comes to some very precise conclusions. So let me first read the abstract. The leading cause of mortality from SARS-CoV-2 is an exaggerated host immune response, triggering cytokine storms, multiple organ failure, and death. Current drug and vaccine-based therapies are of limited efficacy against novel viral variants. Infrared therapy is a non-invasive and safe method that has proven effective against inflammatory conditions for over 100 years. However, its mechanism of action is poorly understood and has not received widespread acceptance. We herein investigate whether near-infrared light, NIR light, exposure in human primary alveolar and macrophage cells could downregulate inflammatory cytokines triggered by the SARS-CoV-2 spike protein or lipopolysaccharide, LPS, and via what underlying mechanism. Our results showed a dramatic reduction in pro-inflammatory cytokines within days of near-infrared light treatment, while anti-inflammatory cytokines were upregulated. Mechanistically, near-infrared light stimulated mitochondrial metabolism, induced transient bursts in reactive oxygen species, and activated antioxidant gene transcription. These, in turn, downregulated reactive oxygen species and inflammatory cytokines. That's the therapeutic pathway. A causal relationship was shown between the induction of cellular reactive oxygen species by near-infrared light exposure and the downregulation of inflammatory cytokines triggered by SARS-CoV-2 spike protein. If confirmed by clinical studies, this method would provide an immediate defense against novel SARS-CoV-2 variants and other inflammatory infectious diseases. Now there's a graphical abstract in the upper right. Let me walk through that slowly. So panel A, there's a near-infrared light source radiating near-infrared photons. They strike mitochondria. That's K1. In response to the near-infrared photons, the mitochondria release reactive oxygen species, ROS. One of those is hydrogen peroxide, H2O2. Let us pause here for a sidebar comment. At first sight, this is surprising. Near-infrared light is supposed to be therapeutic, yet what the authors have demonstrated is that near-infrared light, by striking mitochondria, produces a very rapid release of reactive oxygen species from the mitochondria. That's a key new piece of information. What happens is the cell reacts to this reactive oxygen species burst, which occurred in response to near-infrared light, and it adapted. It turned on anti-inflammatory pathways to deal with the reactive oxygen species. And by doing so, prepared the cell to undergo a shift in its cellular trajectory. Instead of being in a pro-inflammatory state, with repeated near-infrared exposures, the cell, by adapting to the reactive oxygen species, became resistant to the reactive oxygen species. And we will see one of the key enzymes which was upregulated in response to the near-infrared light. The net result of this is that pro-inflammatory cytokines are suppressed, and that's shown on the right, cytokine secretion being suppressed with the X. Now moving to panel B, and that's the lower part of the inset of the graphical abstract. I have covered up B with the picture of catalase, but this lower sequence shows near-infrared photons being delivered to cultured cells for various amounts of time. 5 minutes, 10 minutes, 15 minutes, and 20 minutes. There's a typo in that legend. 1 should actually be 15. And what they're showing is the amount of ROS, reactive oxygen species, is going up in rough proportion to the duration of the near-infrared photons being delivered to the cells. So K2. So once the cells have been dosed with near-infrared radiation, have produced reactive oxygen species in response to that, then they analyze the amount of cytokines in the cells. And what they found was an optimum duration of time to reduce the pro-inflammatory cytokines. And that optimum time was 10 minutes. So once they had decided on using 10 minutes of exposure time for near-infrared irradiation of their cell cultures, they then proceeded to utilize both spike protein as well as another agonist of the toll-like receptor 4, lipopolysaccharide, abbreviated LPS. LPS is commonly used in cell cultures and rodent studies to provoke rapid inflammation. So using the 10 minutes of near-infrared illumination as well as the LPS trigger for cell inflammation, they conducted an experiment which is shown, the results of it, just to the left. And they're looking at CAT, which stands for catalase. Catalase is a key enzyme which degrades hydrogen peroxide. And catalase is primarily located inside peroxisomes, which are often adjacent to mitochondria, which produce hydrogen peroxide. Now that's a lot of verbiage. Let me go to the experiment. They're looking at the amount of gene expression for catalase, not the protein, not its biological activity. They're looking at the amount of gene expression, amount of RNA. So in control cultures, there's a certain amount of catalase production. And this is the RNA which will make protein, catalase protein, which will be stored inside peroxisomes, ready to deal with hydrogen peroxide. So in response to lipopolysaccharide, that's the blue bar in this histogram, note that this toll-like receptor agonist, which provokes cells into an inflammatory condition, depressed catalase levels. And this is potentially bad because with reduced catalase, the ability of the cell to degrade hydrogen peroxide is reduced, leading to conditions that could promote ferroptosis. Now the red bar illustrates the therapeutic benefits of near-infrared light. these cells were treated with a combination of lipopolysaccharide and 10 minutes of near-infrared light. Notice that the catalase levels were boosted in response to this. So the result of lipopolysaccharide is to stimulate toll-like receptor 4, which sets off a pro-inflammatory cytokine cascade. There's also a production of reactive oxygen species in cells in response to LPS. Normally, in the blue bar, those cells, catalase messenger RNA expression was depressed. But in the cell culture, which was stimulated with near-infrared light, the opposite happened. Catalase levels went up, and this again is the messenger RNA, the measure of gene expression. So this deep dive into the levels of catalase messenger RNA expression in response to a pro-inflammatory agonist, which is found on bacterial surfaces, bacterial cell wall surfaces, utilizing near-infrared light, one produces a state which is more advantageous for the cells to degrade hydrogen peroxide, reduce this type of reactive oxygen species, and thus reduce the susceptibility of the cells to ferroptosis. One may ask, how significant is LPS in the context of cellular pathophysiology? LPS is a common component of the cell wall that is beyond the cell membrane of the bacterium of gram-negative bacteria, which include E. coli. In E. coli, LPS constitutes 80% of the molecules on that outer surface, the cell wall of the bacterium. So let me put the context of ferroptosis in a tissue and organismic context. If you have bacteria on the surface of an epithelium, such as the gut or the lungs, and LPS is being liberated, setting off toll-like receptor 4, pro-inflammatory cytokines, reactive oxygen species, if the cells are not able to process the reactive oxygen species, this will lead to peroxidation of lipids, setting the stages for ferroptosis, or programmed cell death. These will lead gaps in the epithelium, allowing the bacteria to penetrate into the deeper tissues of the organ. And this is what's called sepsis. And sepsis is a common pathophysiological state found in advancing progressive COVID patients. Now, shifting the discussion, the narrative back to the COVID patient who was being treated with sunlight. They wheeled this patient who had a pulmonary infection of COVID-2. His condition was worsening. They moved him to sunlight, and within a day, his pulmonary functions improved. And within several days after that, he was discharged. How could sunlight produce such a rapid change? Well, if we look at this paper, which was published in 2023, we go all the way down to the level of catalase messenger RNA being upregulated in response to near-infrared photons. As proteins, catalase proteins, are upregulated, these cells will be able to resist reactive oxygen species more effectively. And that would be of major benefit for somebody who is suffering from a COVID-2 infection of the lungs or the gut, because if catalase is deficient, reactive oxygen species will set off a cascade of results leading to ferroptosis. And that's shown at the lower left of this slide. That needs to be prevented. And this is where sunlight and near-infrared lights now appear to be a promising way of arresting the pathophysiology that's taking place, which could lead to sepsis. And so, again, reading from the abstract of this paper, if confirmed by clinical trials, this method, using near-infrared light, would provide an immediate defense against novel SARS-CoV-2 variants and other inflammatory infectious diseases. So, takeaway concept. We have seen that there is a high consistency of viral attacks that are focused on antioxidation pathways. And some of the key players inside cells that provide this antioxidant capacity are the cysteine glutamate antiporter, also known as XCT, glutathione, abbreviated GSH, NIRF2, the key master anti-inflammatory antioxidative transcription factor, which upregulates genes such as GPX4, glutathione peroxidase 4. And we've seen how researchers emphasize this is an essential enzyme to resist and degrade reactive oxygen species and to chemically reduce peroxidated lipids, which are the upstream drivers of ferroptosis. Now, adding to this list is catalase, the enzyme which is placed in peroxisomes, which are located next to mitochondria. And catalase, its role is to convert hydrogen peroxide into water and neutralize it, neutralize the hydrogen peroxide. Now, the importance of peroxisomes in COVID and long COVID has only become apparent in the last couple of years. I would like to draw your attention to this blog from the Brain Inflammation Collaborative, Oxidative Stress in Long COVID and ME/CFS, Myalgic Encephalitis, Chronic Fatigue Syndrome, the Mitochondrial Peroxisomal Vicious Cycle. Oxidative stress and mitochondrial dysfunction drive fatigue and brain fog in long COVID and ME/CFS. Discover the peroxisomal vicious cycle. Proven ways to boost glutathione, NAC, N-acetylcysteine, coenzyme Q, and NIRF2, boosting NIRF2. And as we've been discussing, one way to boost NIRF2 is with melatonin, which can be produced by near-infrared light.